welcome everyone. Thank you for attending, and also congratulations. You found the right ship. It's impressive. You got yourself here, and this is a pretty special place. So this is the inaugural episode of a multi-part webinar series put on by the American Spinal Injury Association by, with, and for early career professionals. So this is a bench to bedside view of the landscape of spinal cord injury in medicine through the eyes of us early career professionals. And in this multi-part series, we will be focusing each episode on a single topic relevant to SCI research in medicine. And in each of these standalone episodes, a dyad of presenters will present a parallax perspective from two different viewpoints based on their expertise, ideally one being more laboratory focused and one being more clinically focused. Each presentation will be about 20 minutes with exactly 10 minutes of Q&A afterwards. And beyond this sharing of the emergent work of the young professionals in this space, this series also has other intentions. One is to identify and archive these rising stars, and another is to create a community for networking together, all of us people who have a shared common mission. So this is where we are at. First off, let's start with some acknowledgements. First, thank you so much to all the presenters, the hard work that you do, and your science is the only reason we're doing this. Also to the founders and co-organizers, so that includes myself, Dr. David McMillan, Dr. Annie Palermo, and Dr. Raul Sadcheva, and also all of Asia's front office and Asia writ large. And so much thanks, especially to the front office. So huge shout out for all the efforts that come out there. And then a little bit of housekeeping here before we get started with the first presenter for the first episode. So if you have a question for the presenters, please drop that in the chat. If you are willing, after the presentation, we will call on you so that you can identify yourself and ask your question. Just like unmute yourself and ask. If you would not like to do that, simply let us know in the chat and we'll read your question off. So you have both options there. And then afterwards, stick around for a second because there'll be a couple of calls to action as a wrap up. All right, anything I'm missing? No, I'm getting thumbs up. Okay, so season 1, episode 1 of Asia's early career webinar series is on the topic of post-SCI respiratory function. We have 2 speakers. I'll give both their bios now, and then we'll move into the first one. The first speaker is Tatiana Bezdudnaya. I'm sure I said that incorrectly. She's smiling. Good. And here is her bio. So Tatiana's scientific career began with a focus on the role of thalamocortical and brainstem circuits in the processing of somatosensory or whisker and visual systems. This research enabled her to gain an extensive training in electrophysiology, neuroanatomy, and data analyst. In late 2013, Tatiana embarked on an entirely new career path and began studying respiratory neurobiology and plasticity after spinal cord injury. Tatiana's ongoing research interests includes the studying of respiratory circuits, assessing the mechanisms and potentials for respiratory neuroplasticity following high cervical SCI in rats, and developing targeted therapeutic strategies to promote respiratory recovery. The second speaker will be Euan McCaughey. I'm sure I'm pronouncing that wrong as well. So Euan's interests lie in undertaking clinical trials to prove the effectiveness of protocols or technologies designed to improve outcomes for those living with an illness or disability. He has a particular interest in the use of abdominal functional electrical stimulation, or Abdominal Fes, to improve respiratory and bowel function, especially for people living with spinal cord injuries. He holds appointments at the following centers: Senior Research Assistant for Neuroscience Research Australia, Lecturer in the School of Medicine, University of New South Wales in Australia, Associate Dean of Research, Queen's Elizabeth National Spinal Cord Injury Unit, Scotland, and as an Honorary Research Fellow in the Institute of Neuroscience and Psychology at the University of Glasgow, Scotland, where he is now also a medical student. Okay, so we will move on to you, Tatiana, for the first presentation, and if you can just please read the title as well of the presentation.
How can I share my screen?
I believe so.
Let me see. second. Where's my? Okay. Here we go. So the title is Improving Breathing with Lymph Muscle Stimulation After Cervical Spinal Cord Injury. So first, I wanted to say thank you for introduction and thank you for inviting me to give this presentation. And I'm very excited to share with you my research. And so this work is ongoing work, and I will show you how this project was developing and where it's going now. So the beginning quickly about the spinal cord injury. Around 300,000 people suffer from spinal cord injury in the USA alone, with more than 17,000 new cases occurring each year and more than 250,000 cases, new cases worldwide. And what I wanted to point out that most injuries occur at the cervical level, and it means these people will have different degree of respiratory dysfunction. And their treatment options for the spinal cord injury and we researchers working on will include cell therapy, drug therapies, activity-based therapies, and electrical stimulation. So today I will talk about the electrical stimulation, and it could be functional electrical stimulation, epidural stimulation, more invasive, less invasive, transcutaneous, or diaphragm or phrenic pacing. But today I will talk about the lymph muscle electrical stimulation. And question is, why lymph muscle stimulation? So first, we know that there is a functional interaction between locomotor and respiratory network. So when we run, when we jump, when we exercising, it will change our breathing. So we'll start breathing faster and deeper. Animal studies show that lymph stimulation results in entrainment of respiratory rhythm and changes in ventilation. And this was suggested that due to activation of sensory feedback from the lymph muscles. And this is the activation affect the respiratory centers in the brain stem. Also, animal studies shows that there are interaction between locomotor CPG that is located in the spinal cord and respiratory CPG that is located in the brainstem. And locomotor CPG activation results in changing in respiratory rhythm. Also, bimodal neurons were found, respiratory locomotor neurons were found in the spinal cord and the brainstem. And finally, therapies based on repetitive lymph movements and stimulation are becoming more recognized in treating respiratory dysfunction in people with different medical conditions, including spinal cord injury. So I do research using animals, spinal cord injury models with the focus on the diaphragm. So diaphragm is their main muscle of respiration. In particular, this is inspiratory muscle. So you can see it here on the diagram. So diaphragm can be divided in two hemidiaphragm, and each hemidiaphragm is innervated by phrenic nerve. So phrenic nerve is formed by axons of phrenic motor neurons located C3, C5 cervical level. And phrenic motor neurons receive inputs from the brainstem where the respiratory rhythm is generated. So they receive direct inputs or via two prephrenic interneurons. So my animal model is C2 hemisection. So C2 hemisection, basically we do cut the spinal cord, half of the spinal cord, just above the phrenic motor pool. It will interrupt inputs from the brainstem to the phrenic motor pool and result in immediate diaphragm paralysis. So here the example of recording diaphragm EMG from the left and right hemidiaphragm. So you see left and right EMG recordings. So this is the raw data and this is integrated. Each of these bursts, this is where the diaphragm contracting. And this is before C2 hemisection, and this is immediately after C2 hemisection. As you can see, so we also always do hemisection on the left side. So you see there are no any activity on the left side. So we don't see bursts anymore. We see those vertical bars is artifacts, is ECG, electrocardiogram. And also what you can see, the compensation of the right side. So you see a significant increase the amplitude compared to the before C2 hemisection. Okay, so going to the lymph muscle stimulation, we chose forelimb and we chose the biceps brachii stimulation. And we wanted to see the biceps brachii stimulation, how it can affect the respiration on in situ hemisected rats. So for this experiment, we use decerebrate rats and terminal procedure. So decerebration allows us to avoid anesthesia, using anesthesia, because anesthesia will significantly affect the respiration in these animals. Also, we used acute situ hemisection. So we took the animal, dissected phrenic nerves, did the decerebration and situ hemisection, let animal to recover for several hours. And then we recorded phrenic nerve activity on the right and left side, and we used so-called closed loop bicep stimulation. So we were driving the stimulation of the biceps by recording the right or contralateral phrenic nerve activity. So how it looks like, basically, so here you see phrenic nerve activity right, the not injured side, and this is the left, several hours after situ hemisection, so you don't see any activity on the left side. Here, the integrated activity from the left and right side. We start the bicep stimulation, so we do the bicep stimulation in the beginning of each breath in the right side. And you see that during the bicep stimulation, we see significant increase in activity of the left phrenic. Before it was practically complete paralysis, and now we see activity. So this is the average traces to this contralateral side, ipsilateral, the injured one. So you see before there was almost nothing, and during stimulation, significant increase activity. So we were very excited with these results, and we submitted the grant to Cure Paralysis Now and suggested to use closed loop bicep stimulation to treat diaphragm paralysis after situ hemisection. And by condition of this grant, we need to do experiments in chronic spinal cord injury. So we proposed to start to do stimulation at 6 weeks after situ hemisection, do bicep stimulation for 2 weeks, 5 days a week for 30 minutes. So we learned how to do to implant the diaphragm EMG electrodes, how to implant the bicep stimulating electrodes, and do closed loop bicep stimulation. And this is the example. So here you see recording of the contralateral diaphragm EMG activity. And so this is our stimulation. So we drive our stimulus in the beginning of each the contralateral diaphragm EMG activity and stimulate the biceps. And this is how this look like in rat. So you can see there, so first of all, what you can see that rat's breathing is much faster than in human. Also, it's very heterogeneous. It could be slow breathing, it could be fast breathing. And actually, during our stimulating sessions, rat could sleep, could walk around, and do many things. So so we did this in rats, and I found the 2 big problems with my models. So first of all, when we started to do chronic recordings after situ hemisection, we found that there is no long-time paralysis of the hemidiaphragm after injury. So basically the diaphragm can recover in terms of the amplitude, diaphragm amplitude. So here the results from 8 animals. So here we see the diaphragm amplitude during slow breathing in rats before and after situ hemisection. So before, and we compare those after situ hemisection amplitudes to the pre-injury level. So pre-injury 100%. So you see that one day we almost have no activity on the left side, 1 week, and at 2-3 weeks after injury, the amplitude, diaphragm amplitude, practically recovered. So you remember I wanted to do the stimulation of the bicep starting at 6 weeks, from 6 to 8 weeks. So it means that I couldn't use the diaphragm amplitude as my outcome marker of the effectiveness of my treatment. And also here, I just wanted to mention quickly, this is the diaphragm amplitude on the contralateral side. And you see that it's significantly higher than pre-injury levels. So this is the compensation, and then it kind of like going down. And this is the just raw data recording in naive animal, 1 day post-C2 hemisection, 1 week and 8 weeks. Okay, so this was the first problem that I found. The second problem was I found that anesthesia can significantly affect activity of recovered diaphragm. So, for example, if you anesthetize animal, so isoflurane, and put animal in a cage connected and start recording, you might see no activity on the left side or ipsilateral diaphragm EMG. And then 10 minutes after, when animal wake up, you see good activity on the ipsilateral side. So this is recording from the same rat in the same file and just like 10 minutes apart. So in the beginning, I thought is the kind of mistake, and I was checking my connections and trying it on different animals, and I was getting the same results. And then I decided, okay, let's try another anesthetic. So another anesthetic that we use is ketamine xylazine. We use for the long time anesthesia to do surgeries. And basically, I got the same results, no activity on a left diaphragm EMG. And so this recording from the same animal, they're like few days apart. And obviously, so this effect, it's a dose dependent. If the animal start waking up, you might start to see some activity on the ipsilateral diaphragm, or low dose of xylazine ketamine also will give you some small activity on the injured side of the diaphragm. And another way how to assess the recovery diaphragm recovery is the recording phrenic nerve activity in uretan anesthetized animals. So in this case, I was maybe a little bit more successful. So I could see some activity on the left side, the phrenic nerve, left phrenic nerve on the uretan, but its activity was significantly suppressed compared to the contralateral side. And I decided to do this recording and assess effectiveness of my treatment. And unfortunately, so I had 7 rats without stimulation, 6 rats with the stimulation, and I compared the left phrenic nerve activity on these 2 groups animals. So this is the non-stimulated, this is with the bicep stimulation. You see the trend with the bicep stimulation, left phrenic was a little bit higher, but it's not significant differences. And when I compared left phrenics in stimulated versus non-stimulated groups during respiratory challenges, I didn't see there significant differences. So it gave me a lot of thoughts about the model and about this type of the treatment. And one of them was that maybe the protocol of the stimulation is not optimal, and I can try different ways to change those protocols. And one of the ways, this is a researcher for me, is try to understand the mechanisms, how this interaction between the forelimb or the biceps muscle happening with the phrenic motor circuit. So I wanted to try to understand the mechanisms of this. And so recently we got the grant from Pennsylvania Department of Health that basically designed to answer this question. And so this may be a little bit complicated schematic, showing the way how their stimulation of the biceps can modulate respiratory activity. So it could happen through their supraspinal pathways, so their sensory afferents going to the brainstem and can affect the brainstem respiratory CPG. And also it can happen through their local spinal circuits. And in this project, we are very interested or focused mostly on these spinal circuits. So we want to understand how we can activate those spinal circuits, or interaction between biceps activation and the phrenic motor circuit, because it becomes very important after spinal cord injury when the brainstem inputs to the phrenic motor neurons are limited due to the injury. And so we are very interested in those interneurons that allow to integrate the biceps and phrenic motor circuits. And one way to study is to using tracing studies with the PRV. PRV is the pseudorabies virus. This is transsynaptic retrograde tracer. So it can jump from synapse to synapse and basically trace all the circuit. And also it can have a different fluorescent hug. So we injected green PRV into the muscle, left muscle, and applied the red PRV on the left hemidiaphragm. Waited for 3 days, sacrificed animals, and did immunostaining to enhance red and green fluorescence. And so here the example of, and look for the double labeled interneurons. So here you see example of the 2 red interneurons that were traced from the diaphragm. This is 3 interneurons traced from the biceps, and we see 2 double labeled interneurons. So we call these interneurons shared interneurons between biceps and diaphragm motor circuits. And also I wanted to point out, so this is 2 interneurons were found close to the central canal. So this is the, I don't know if you can see it or not. So this is the central canal. So the next step would be to try to identify what kind of these interneurons. And the easiest way, and they're the best antibody that I know, is a CHAD, anti-CHAD antibody. And so we did triple immunostaining for the red anti-RFP, anti-GFP, and the CHAD, and we found a lot of triple labeled interneurons too. And so here we can see the map. This is the map of distribution of shared phrenic biceps interneurons. So this is the very preliminary, not final, and this is more, it's not quantitative, it's more qualitative map. Basically, what I wanted to show here, that you can see, find those shared interneurons from C2 to T2 spinal cord. Also, you can find those shared interneurons not only on the ipsilateral side where we had the traced those circuits, but also on the contralateral side. So here you can see the red is there showing the place of the location of the phrenic motor pool. Green is showing the location of the biceps motor pool. Blue is the triple labeled interneurons. Yellow is double labeled interneurons. And what is striking, I see that the most interneurons, those shared interneurons we see are located in lamina 7 and 10, so they're mostly like kind of like around the central canal. And there, it's kind of like remind me those interneurons, so-called V0C interneurons, those acetylcholinergic interneurons that been shown have important role in locomotion. So it seems that it's only also they can modulate the phrenic motor neurons too. And so the next step will be to try to do triple immunostaining for another neurotransmitter, and because I believe there would be some GABAergic or glycinergic inhibitory interneurons too, those among those shared one. And also we're interested to look for those double labeled interneurons in the brainstem, because we also interested in those supraspinal interconnections between the biceps and phrenic motor circuit. And so next what we do, we record single neuron recording, like extracellular recording within the spinal cord, respiratory neurons, and see how the bicep stimulation can affect activity of the cells. So here the 2 examples of inspiratory cells, and 0 is this beginning of inspiration. So particular this one cell increased the firing rate during the stimulation. This one changed the latency. And I want to say that not all cells are affected by bicep stimulation. Also, we do recording in spontaneously breathing animals and doing bicep stimulation and looking how the airflow changes. So you can see this bicep stimulation changes the airflow. And we're trying to find the best parameters for the bicep stimulation to activate the breathing in naive animals and also in animals after spinal cord injury, C2 hemisection. And also wanted to show you the piece of data that the piece of evidence that proving that there are those local spinal interconnections, pure spinal interconnection between biceps and phrenic motor circuits. And to see it, we need to remove all those supraspinal inputs from the brainstem to the phrenic motor neurons, and this the one of the way to do it is do complete C1 transection. So this example in C1 transected threads, and also we believe those intraspinal interconnection in the normal conditions, kind of like they are suppressed, inhibited, and the one of the ways, this way to disinhibit those interconnections is using blocker to intrathecal delivery gabazine strychnine. So they are the blockers of the fast inhibition. But so we apply the very low dose of these drugs. We don't want any like sporadic or like movements of the animal when everything is disinhibited in the spinal cord. So basically what you see here, this is the blood pressure of the animal, C1 transected animal. So this is the tracheal pressure, and the animal was under mechanical ventilation. This is end-tidal CO2 level. So, and we start to do the bicep stimulation. So you see this, the bicep stimulation, and turn off the ventilation. And you can see the animal started to breathe with each of the stimulus of the biceps. And you see this end-tidal CO2 kind of like it's higher because obviously it's not normal breathing. But for me, it's the evidence about those pure interspinal interconnection between the biceps and the phrenic motor circuit. And then my last slide, maybe a little bit more complicated. So we are collaborating with the team from Dr. Ilya Rybak and Natalia Shevtsova. They work in Drexel in our department and doing computational modeling studies. So they have a nice model of the respiratory system at the brainstem level. So we want to include the spinal cord level. So this is the kind of like preliminary model. And we also want to include these sensory inputs to the brainstem and to the spinal cord from non-respiratory muscles. And we also want to be able to simulate the respiratory deficit following spinal cord injury. So basically, we can simulate C2 hemisection when we remove all those inputs from the brainstem to the spinal cord, so remove all these inputs, or complete transection, remove all of these inputs, and do, like, let's say, the stimulation of this the non-respiratory muscle afferents. And so we believe this model in all our study will allow to find the best protocol for the bicep stimulation that can be translated further in a human population. And so here are my acknowledgements. To thank you for my collaborators, Ilya Rybak, Shevtsova, Vitaly Marchenko. So he was helping with the terminal recordings, Michael Lane, and lab members that they're helping me with the tracing now studies, and to my former and the current funding support. And I guess that's it. Thank you. Thank you for your attention.
Thank you so much. So if you can stop screen share, please.
Mm-hmm.
We have one question from Jessica. We're a bit over on time. So Jessica, if you want to unmute yourself and ask, please do so quickly.
Hi, I just wanted to ask, is there evidence that the diaphragm shares excitatory interneurons with other muscle groups in humans?
I don't know. There in humans, it's not known, but as I said, there are some studies or like the repetitive other stimulation of their sensory feedback from the muscles is used to activate like respiratory activity in the humans, like how to say it. So So there are not direct evidence, and yeah, so I don't know, maybe. So first we want to show it in the animals first, but I don't think so. There are real evidence in the human, but yeah, this is what probably my response.
Sounds like we have to find that, figure it out.
Yeah, yeah.
Thank you.
I guess there are. I think there are, because I think it's the basic how the central nervous system is composed. So we have kind of like the systems, and all the systems should interact somehow, and those interactions are kind of like they have the rules to interact between each other. So I believe there are those shared interneurons in the human too. Yeah.
To both, this is actually related to a question I wanted to ask that maybe we can bring up at the end if we can pose some more questions after, just in case anyone has to leave at the scheduled end time. Okay, Euan, if you could queue up your slides and begin now.
Sorry about this, guys. Let's just go this way. Thanks for the introduction, guys. My name's Euan. Thanks very much to the Asia group for inviting me along. I guess there are pros and cons to a global pandemic, and one of the pros is that I get to talk to you guys in my pajamas at 10 o'clock at night on a Friday. So I have been asked to give a bit of a talk about my research, but also to talk a wee bit about my career trajectory, how I got here and stuff. So look, some of the graphics probably aren't going to work all that well here, but if I was doing this talk face to face, I would probably wouldn't be using this slide deck anyway. So I'm currently based in Glasgow, but I'm running my research lab in Sydney, Australia. We'll explain a wee bit about how we got here. A quick conflict of interest statement before we start. A lot of the content around abdominal stimulation here will allude to equipment developed and commercialised by a company in Kentucky, America. I've got no affiliation with that company, and I've got no conflict of interest. It's simply the best gear for the job at this point in time. So who am I? What have I done? So I guess I would now probably be classed as a neuroscientist or a physiologist or something, but my background's actually in mechanical engineering. So in my master's year of my mechanical engineering degree, I ended up undertaking a control engineering project looking at systems to improve respiratory function in spinal cord injury. That was now 11 years ago, and I've ended up staying in that space ever since. I then conducted a biomedical engineering Ph.D. in Glasgow, where the focus was primarily spinal cord injury and techniques for that, and just general improvements in respiratory function. A quick postdoc at the University of Strathclyde in Glasgow, looking at electrical direct current stimulation of the spinal cord, before I headed out to Australia to get away from the cold and the wind and the rain in Glasgow. I bounced about a couple of places there before landing at Neuroscience Research Australia, where I started off as a collaborator in a group, and within a couple of years I am now a PI, running a research group who focus on the clinical translation of medical devices and algorithms and paradigms to improve all sorts of functions and outcomes in spinal cord injury and critical illness. And Annie is one of my postdocs. So I'm currently running that group remotely a day a week. I'm also the Associate Director of Research for Spinal Injury in Scotland a day a week. And in September of this year, due to the pretty much dire funding situation in Australia, wants for a new challenge and things, I have decided to go back to university, and I am currently undertaking an undergraduate medical degree at the same time as trying to run my research group and be the Associate Director of Research in Spinal Injury. So it's a pretty busy time. Sometimes get to see my wife and kids, but enjoyable and fun all the same. So that's a wee bit of context about how I got here. I just want a quick slide here about respiratory function and spinal cord injury. Look, I'm sure nearly everyone here is aware, but obviously the diaphragm, the intercostal muscles and the abdominal muscles are all innervated at different levels of the spinal cord. And typically almost all people with spinal cord injury who will see will have some sort of respiratory muscle impairment. That may only be the abdominal muscles, but once the injuries get a bit higher thoracic, it might include the intercostals. And once we're getting to the C3, C4, C5 area, we'll also have phrenic nerve and diaphragm paralysis. And just to kind of highlight the severity of that, put together a wee table here where we're looking at the a number of respiratory function measures in people with tetraplegia compared to the percent predicted for the able-bodied population. And you can see that these numbers are starkly lower than the predicted values. Now, why that's relevant to me in my research group is that these low numbers will typically correspond to a number of clinical side effects, such as repeated respiratory complications, some people requiring intubation and stuff, which is going to be the focus of our talk. How can we improve these outcomes and effectively improve quality of life and well-being for spinal cord injured patients? So maybe if I was doing this face-to-face, I would ask for a show of hands, but maybe by a thumbs up in the chat. I just would like to know how many people have used measures with spinal cord injured patients to improve respiratory function. Now that can be respiratory muscle training, it can be electrical stimulation, whatever, but that will give me a bit of an idea of what the audience is as well. So if you can stick a thumbs up if you've ever used any respiratory muscle training. Okay, so I've only got a few people, so that's good. I'll keep things pretty basic then. And give this as a bit more of an introduction of how we can use it, why we can use it, etc. So, as I said, some of these slides aren't going to look particularly great. One of the interesting areas that in Australia is respiratory muscle training and spinal cord injury. Now, I'm only going to allude to this briefly, but what it effectively means is breathing against a resistance, so that you're effectively exercising your respiratory muscles, so primarily your diaphragm, and by repeatedly breathing against this resistance, you should begin to improve your voluntary efforts. Now, we had a nice paper published in, I think it was Thorax last year, where we undertook a clinical trial with 62 adults with tetraplegia, and they undertook respiratory muscle training or a sham twice daily for 6 weeks, and the intervention was effective. We got a good improvement in maximal respiratory pressure. People said the respiratory symptoms were reduced, and nearly every patient who got the intervention reported an improved quality of life. But as you can see on the screen, there's a big but here, and the interesting but is we then left these devices with the patients for another 6 months. So we said, here is your research device. And for all those people who got the sham, we gave them the real one. And we said, keep using it and we'll follow up with you again in 6 months. Now, the training in the clinical trial was supervised. The training for the following 6 months was not. And if someone wants to post in the chat a rough guess out of how many of the 62 adults continued using respiratory muscle training, despite them reporting improvements in respiratory symptoms, quality of life, and us saying that your respiratory function was better. So on you go, have a guess in the chat. Anyone willing to hazard a number out of the 62? So someone said 62, someone else has went for 60. You're very optimistic. The actual number was 2. 2 of the 62 people, once the intervention became unsupervised, continued to use it. So one of the hypotheses for this is people only really regard respiratory function as being important is when they're not well. Once they're actually well, they go, well, I've got other things I need to focus on. The rest of the focus of this talk is going to be on electrical muscle stimulation. The nice thing about electrical muscle stimulation is it's relatively, or abdominal muscle stimulation is it's non-invasive. People can train at home when they're watching the telly, doing other things, etc. Let's go into the main body of the talk. I'll probably only talk for about 10 minutes. So there are three main respiratory muscles that we can target with electrical stimulation. For the diaphragm, we can use phrenic nerve pacing or diaphragm stimulation. Now, these are for your very high-level, complete spinal cord injured patients, typically people who would otherwise require a mechanical ventilator. Phrenic nerve pacing normally involves directly stimulating the phrenic nerve in the neck, whereas the new technology is diaphragm stimulation, where you use a laparoscopy to place the electrodes directly onto the neuromuscular junction on the diaphragm. Now, both of these techniques are fine and well for people who require a mechanical ventilator, but they represent a very small percentage of the spinal cord injured population. They're very expensive and they're very invasive, so I'm not really going to touch on them any further, but I'm happy to answer any questions about them later in the chat. Intercostal muscle stimulation is an interesting concept, but the intercostal muscles are interwoven. You've got both inspiratory and expiratory roles of the intercostal muscles, making them very difficult to reliably stimulate electrically. So that leads us on to the abdominal muscles, and the abdominal muscles are much easier to stimulate compared to the other groups because they're a big muscle and they're easy to get at from the spine. So there's a couple of techniques that have been used. Spinal cord stimulation involves implantation into the spinal cord to improve the abdominal muscles. A lot of that work's been pioneered by Anthony DeMarco in Cleveland. However, it's a relatively invasive technique, and we were left thinking, well, the results are great, but can we achieve that non-invasively? There's two ways to do that. Magnetic stimulation involves using a large magnet placed over the back of the spinal cord to activate the abdominal muscles. You can achieve cough, deep breaths, whatever. The only downside with the magnet is it's very large, it gets very hot, it's bulky, and it's expensive. This kind of left us going, well, the other option is abdominal functional electrical stimulation. What can we do with it? Now, these slides are a bit cluttered because the slide hasn't worked all that well. However, abdominal muscle stimulation is the electrical stimulation of the external obliques and or rectus abdominis muscles, and I'll talk about the actual stimulation protocols in a minute. However, the nice thing about abdominal stimulation, as I said, it's completely non-invasive. You're stimulating over the surface of the muscles. And in a meta-analysis shown here, we've been able to show that abdominal stimulation instantaneously improves cough peak flow and a number of other respiratory outcome measures. That is, as soon as it's turned on, when you breathe with the stimulation, you improve respiratory function. Improved cough means better airway clearance, reduced respiratory complications, etc. However, it doesn't always correlate to a person improving their own unassisted breathing. That same meta-analysis was able to show here that there is also a training effect of this stimulation. So as you can see, the black bar represents when people got abdominal stimulation training, that is, they used it every day, and this shows their own respiratory function. You can see that it improves quite greatly. And finally, we've also been able to show that this technology may reduce mechanical ventilation duration. So I'm going to skip on to the next slide. The electrode placement that's been used is very varied. Now, as you can see here, we've got 7 different placements with generally a lack of consensus as to the best electrode placement. However, we undertook a study 2 years ago now in Sydney, where we used a number of different electrode placements, and we were able to show that this position, which we call the posterior lateral electrode placement, generated the highest intra-abdominal and esophageal pressure during the electrical stimulation. Now, high intra-abdominal and esophageal pressure is what you want to good cough, loosen the mucus, etc. in the airways. So the rest of this talk will focus on this electrode position, and this is the electrode position I would recommend be used clinically. Now, respiratory function is the primary mechanism we're targeting. However, as I said, respiratory function is actually a pseudo measure of what we want to achieve. What we actually want to achieve is better clinical outcomes for our patients. However, you can see from these graphs here where a patient has been asked to undertake a voluntary and stimulated cough. You can see with the gastric pressure and esophageal pressure during stimulation, which is this black bar, it's a massive difference in pressure generation thanks to electrical stimulation. Now that looks nice enough in the picture, the flow volume looks lovely, etc. But what I'd like to actually do for you here is share what it sounds like, because I think this gives the best impact. So what you're going to hear, first of all, here is a patient taking an unassisted cough. And then secondly, you'll hear stim on, and that's the cough with stimulation. And the difference is striking. So hopefully this works.
Are you ready, Ryan, and cough? Good. Are you ready, and stim?
Okay, so I'll let you run through that again now that you know what to expect. But as I said, the first cough is unassisted, and the second cough is with stimulation.
Are you ready, Ryan, and cough? Good. Are you ready, and stim?
And we can see with the flow volume loop here that some of the expiratory flows are actually getting towards near normal values, and this is a C4 complete tetraplegic patient. So obviously the stimulation is doing something. Now, as I said, we undertook the meta-analysis that showed abdominal FES led to an acute improvement in cough peak flow. What's actually hiding under this graph here is that the abdominal FES training was also found to lead to a significant improvement in unassisted vital capacity, forced vital capacity, and peak expiratory flow. That is, if you train for long enough, you improve your own measures of these breathing functions. Now, what does that mean? Well, if we consider mechanical ventilation duration first, around 40% of our tetraplegic patients require mechanical ventilation duration at some stage of injury. And we've shown that for a patient who was 9 months post-injury but unable to be decannulated due to repeated respiratory complications, daily abdominal FES had the person breathing independently after about a week, and after 3 weeks they were decannulated and they never were intubated again. So it was almost that the abdominal FES was that small stimulation that had to get them over the threshold to breathe independently. In a separate study, we've also demonstrated that 5 times weekly abdominal FES applied every second week led to what appeared to be faster weaning from mechanical ventilation. And unrelated to spinal cord injury, a recent pilot study we conducted in Australia with general ICU critically ill patients showed that abdominal stimulation led to a statistically significant reduction in ventilation duration and ICU length of stay for critical ill patients. So I think we're now in the right ballpark. We're now getting this technology that showed an physiology-based improvement and now leading to improved clinical outcomes for our patients. So that poses the question, what next? So we're currently undertaking a study looking at respiratory complications in spinal injury. So respiratory complications, as you'll all be aware, are a leading cause of illness and death for people with tetraplegia. And in the first 6 weeks of injury, Jackson and Grooms reported, albeit about 25 years ago now, that around 68% of people with tetraplegia are experiencing some sort of respiratory event in the first 6 weeks post-injury. So a reduction in respiratory complications would also obviously have great outcomes for these patients, because we now know that the number of respiratory complications experienced during your hospital stay is directly related to lifelong measures in spinal cord injury, such as number of rehospitalizations, such as hospital length of stay, and things. So we're undertaking a study where within 5 days of tetraplegia, we're trying to start applying abdominal FES. And abdominal FES is applied for 45 minutes a day, 5 days a week for the first 6 weeks of injury to try and get people through the danger zone. Now that study is fully powered and requires 240 participants. We've got 65 participants recruited to date. As I said as well, ventilation duration is a large problem for the tetraplegic population. So as a sister study to the respiratory complication study, where in the respiratory complication study, we're now not taking people on a ventilator because they've got much higher risks of respiratory complications at baseline. We're now putting them onto a mechanical ventilation study where people will receive stimulation for 45 minutes a day, 5 days a week, for as long as they remain in ICU. That study is relatively new with only 4 participants recruited to date. But as you can see, we've taken this technology from a very physiology component to now very clinical. Just going to spend the last minute talking about a couple of other applications. We've got anecdotal evidence that abdominal FES may improve bowel function in spinal injury. Now, that anecdotal evidence says that as soon as you turn on the abdominal FES with a lot of our patients, the pressure builds up and they have a bowel movement. That can be within the first 2 or 3 minutes of applying the stimulation. And that can be, the interesting thing about that is, for some of our studies that we run 5 days a week, it will happen to the patients Monday to Friday, instantly almost. They then won't move their bowels all weekend. We'll come in on a Monday morning, turn the stimulation on, within 5 minutes they've moved their bowels again. So there's definitely something happening there. We're undertaking a study in the chronic population, looking at whether abdominal FES applied 3 times a week for 6 weeks reduces whole gut and colonic transit time. And that uses a nice device called the Smart Pill, which is a device you swallow. It measures temperature, pressure and pH throughout the gastric tract, and it can effectively tell us how well the bowels are functioning. We've also noticed that abdominal FES seems to improve blood pressure control for very hypotensive patients. Whether it's just the stimulation is causing some sort of reordering of the system, whether we're actually causing what we'll term good autonomic dysreflexia or something. However, for the hypertensive patients who can't get out of bed, this technology seems to raise their blood pressure to a level that they're no longer dizzy. They can be brought up, they can be hoisted, they can be transferred, but that's not really the focus of this talk. Finally, a quick plug for Mura, but I'll skip over that in interest of time. There's my funders. I've also put my Twitter handle. I'm happy to take any questions over Twitter that people might have cropping up after this talk. I'm also able to take any comments, questions just now, and I apologise for the slides not working, but I don't think we've really missed out too much.
Thanks, Euan. If you don't mind making me a co-host again, or you can make me the host if that's okay. Thanks for that talk. And I just quick before we get to any questions, I want to let people know that we are going to be having another one of these webinars next week. And Euan, thank you for bringing up the abdominal FES with its impact possibly on bowel function, because that's the topic of our next week's session. We'll be talking about bowel and bladder function. And I think actually one of our presenters, if he isn't still here, he was here. Is Arju still here? Maybe? No, that's okay. But anyway, thank you, Euan. And does anybody have questions? Oh, it looks like we have one. Reza, if you'd like to ask your question, you can go ahead and unmute yourself.
We were just wondering, we're the UNM residency service that's listening in on the lectures. Thank you for a great lecture. We were wondering, is there a BMI limit for the use of the device?
We try not to set a number, so I don't want to get into too much of the clinical application, but I'm happy to discuss it now. So what we do is we effectively stick the stimulation on, and we're not really guided by any quantitative number. What we're looking for is a strong visible contraction of the abdominal muscles. Now that can occur for some people at as little as 20 milliamps, at some people it will be much nearer, up to 100. But devices tend to be limited at about 110 milliamps. As for the use of BMI, it will depend on a number of other factors. It depends on subcutaneous fat in the abdomen. It depends how hairy a person is, so the contact you can get in the skin. But I guess a general rule is if a person's getting to a BMI over 40, especially with a spinal cord injury. So the spinal cord injury tends to mean that people require a higher level of stimulation than they would if they were able-bodied. For let's say a tetraplegic complete injury, a BMI over 40 is going to become difficult to stimulate them. That's not to say it's not possible, so I would never like to put an absolute number on it, but you'll quickly be able to see when you're actually using the device, this is never going to work here. And don't get me wrong, there have been more. I've used this technology probably in around 200 people, and I would say that I would probably be on maybe my third hand now for the number of people who we've not been able to stimulate. So it's not non-existent, but I would say it's definitely less than 10%, probably less than 5%. Thanks.
Thank you.
Quick clinical follow-up question. Can you briefly comment on the accessibility of the electrodes that you use for the abdominal FES approach you described?
Sorry, by accessibility.
Can I purchase them commercially? They look like they're long and rectangular.
Yeah, so those long rectangular ones are actually cheating somewhat because they're just the standard 5 by 10 centimeter axle guard ones. But we managed to find a thing on Amazon, and I'm happy to share the link with people if they email me or tweet me, that effectively lets you apply those electrodes in parallel. So while we've only got 2 channels coming out of the stimulator, you can use these. I don't even know what they call them. We just stumbled across them by chance, but you can wire all the electrodes together and you create a parallel circuit where every electrode's got the same current. So rather, someone asked me the other day, actually, they emailed me asking what size of electrode do you recommend, and that will just depend on the patient. I like to cover as much of the space between the top of the iliac crest and the 12th rib as possible, and that will vary. Now, I told you that there's a company in Kentucky who have now got commercial or they've got FDA emergency therapeutic use for this device. They've also got CE marking, so it can be used in Europe and the rest of the world. Now, one of the neat things about that device, I should say that all the abdominal stimulation we use is synchronized with respiratory function. So the abdominal muscle is only active during exhalation, so we only apply stimulation during exhalation. This device from the group in Kentucky automatically synchronizes the stimulation with exhalation using a nasal cannula. And the other thing that they've done that's quite neat, and it's the first time I've ever seen it, they've made around a 14 centimeter long electrode, but it's got 3 tear-off strips. And that doesn't affect the integrity of the electrode. The concern before with doing things like cutting electrodes was that you would always have a large charge build up in the corners, and that's where the burns might occur, etc. But these use very neat tear-off strips, so it's effectively a one-size-fits-all electrode. And look, the device isn't cheap; it's probably around $2,000 for the whole system. But the device itself can be used with multiple patients, and the electrodes themselves are only about $20 a packet.
So unless there's any other questions, oh, Rahul, go for it.
Wonderful talk. Thank you so much. I just had a question about the injury severity and an inclusion criteria in your studies. And this is also sort of to loop in the first presentation. Can you make a comment on the spontaneous recovery after spinal cord injury? And will that be a challenge in human studies like it was in the animal studies?
I'll answer quickly. From my point of view, we don't exclude anyone. We have to be aware that the incompletes, so the HSCs and Ds, will be able to feel the stimulation. However, they never normally report it as painful. It normally gets reported as pins and needles or some other similar sensation. If patients are reporting it as painful, your stimulation is too high, because the muscle receptors should be coming on before the pain receptors are. But again, happy to answer questions like that over Twitter or email. Tatiana, I'll pass.
Thank you.
Tatiana, do you have any thoughts there?
Okay, yeah, so it's the big problem with the animal studies, the models of the spinal cord injury, because so with the respiratory system, for example, we cannot injury rat so much and keep it on mechanical ventilation for several days. So it's unhuman and it's impossible. And this is happening with with many spinal cord injury patients. So that's why injury should be enough for animal to survive. And there is a lot of spontaneous recovery too. So that makes our work difficult. So it's easy when you do, like, for example, locomotor system, you do transaction, you have movement, handling movements or not, it's 0 or 1. This respiratory system, it's not like this. It's always somewhere between 0 and 1. And also there are so much spontaneous recovery, and it's very difficult to assess if this really treatment worked, you know. So this is kind of like my input.
Thank you very much.
Mm-hmm.
Okay, given that we have exceeded time, I mean, I feel like I have 3 hours worth of questions for everyone. We can definitely continue this conversation, hopefully online. Please, find us on social media. I think I'm going to do a little thread about part of what was alluded to with Tatiana's presentation, having to do with interappendicular neurological coupling between the upper extremity and lower extremity limbs, and why that might have some tie-in to the respiratory circuits. But yeah, thank you everyone so much. Thank you so much to the presenters. Thank you to the organizers and all of you, the attendees. So quickly at the end, if you're an attendee and you have an exciting project you'd like to tell us about, please consider submitting. So each of the seminar series will be on a topic. The topics are listed in the submission form, so you can find it there to see if your topic qualifies. Most likely if it does, and if you think it doesn't, please just reach out to us an email. If there are other organizations or groups who share this mission of platforming and highlighting early career researchers and clinicians in the spinal cord injury space, please let us know, reach out to us, and we can collaborate to make it so that all of this happens better. And then finally, oh, nope, I already said that. All right, thank you all so much. It was an absolute pleasure, and we will see you all next time.
Thanks, guys.
Thank you.
Thank you.
Thank you.
Thanks, everybody.